Everyone, my name is Ella Shiratsky, and this is a tutorial about how to use a tool called BBMap, which is part of the BB Tools suite, to map reads to your reference genome or genomes. So the way BBMap works is it uses a scoring algorithm to determine how your read matches a certain area in your reference genome. How well the read matches depends on how many matches, how many mismatches, and how many indels it has. One of the advantages of BBMap is it can handle really large indels, so it should be suitable for working on genomes, eukaryotic genomes that have large spliced areas. So you could have a part of your read on one exon and the other part on another exon, and it should still map properly to the gene. The way this score is calculated within BBMap is a little bit non-trivial. So you can decide what kind of threshold you want to use to pick whether or not a read maps to a certain area in the genome. But the ratio is a little hard to calculate because it really is a measure of what's the score the read got versus what is the best score that it could have gotten if it was 100% matching to that area in the genome. This parameter where the ratio is called min ratio. So if we run BBMap help, it has a lot of parameters that you can set, obviously. The default minimum min ratio that is set by BBMap is equivalent to 76% ID match between the read and the reference without any indels. So if you wanted to get a better confidence of mapping, because 76% is really not all that great, especially if you're dealing with genomes that aren't spliced, so you're not expecting a lot of indels, you can set instead a parameter called min ID right here. So like you see, the default is 76%. But of course, we're going to set it higher when we actually run BBMap ourselves. The one other thing that you want to consider is BBMap by default will use all of your available threads. If you're just working on your own personal computer, that's fine, just leave it as default. If you are working on a shared machine like a server or whatever where resources have to be split between multiple people, you might want to set this parameter threads here to whatever is the limit of number of threads you can use. Here, this is the binder, so we only have one thread, so we're not going to use this parameter, but please be aware of this if you're using a shared machine. All right, so let's see how BBMap works. If we look at our data, there is a reference genome here, and there are 4 read files, sorry, here at the end that start with SRR that can be mapped to this genome. These are just files that we're going to use today. Of course, you can have as many files as you want. All right. So let's start with the simplest use of BBMap, and you can follow along this tutorial if you look in Topic Metagenomics Lesson 4, you have an SH file that's a binder batch file. So you can run all of it, or you can just follow along in there, and it's going to have all the commands that I'm going to use. So the simplest way to use BBMap is BBMap ref tells you what's the reference. So we're going to look into data and DVH, and you do not need to unzip any of the files that you're using here. So that's a really big advantage to BBMap. We have paired reads here, so we can set paired read file 1 or the forward. So let's start with this sample. And then the second file or the reverse read. Okay. So what we've told BBMap so far is we have a reference genome and we have 2 read files that we want to map to it. The next thing that we need to tell it is the kind of output that we want. So I like to use this parameter called outm. Please note the m here. What outm will do is it will only write the reads that actually mapped to the reference genome into your output BAM file or SAM file, whatever you want to use, but BAM files are more space friendly. So the importance of using or writing only the mapped reads to your output is that your output file would be a lot smaller and take up a lot less space on your personal machine or your server. So that's why I like using it. So I am going to call this DVH and then the name of the sample, mapped.bam. And finally, once again, you can use the default min ratio or min ID. I like to set it a little higher. So what I'm doing now is I'm telling BBMap to only write reads into the output that mapped at least 95% ID. And BBMap knows how to convert that to a min ratio. So if we look at the output here, it's going to say set minimum alignment score ratio to 0.908. So this is a translation done by the BBMap algorithm to what it uses to tell whether to read maps or not. Some other stuff that you can see here is it's using 1 thread, and it'll keep a score of the time that it takes to do everything. This is not going to take a long time because we don't have a lot of reads here. Should be done in about a minute. So in the meantime, I can start talking about what we're going to do after that, because this is the simplest case, right? 1 reference genome and 1 set of forward and reverse reads. But what if you have more than 1 set of reads that you want to map to your genome, right? That's usually the case. Like if you're running an experiment on a genome in a culture under 2 different conditions, then you're going to have reads, 2 separate sets of reads. And we're going to get to how to do that in a second. But first, I want you to look at the output that BBMap gives you. So it took sorry, it's supposed to say how long it actually took. 75 seconds to map these about 200,000 reads per file, so 400,000 reads to our reference genome. So that's pretty fast, which is another thing that I really like about BBMap. So let's look at the output. So we have 1 genome, right? And the maximum indel that BBMap allows by default is 16,000 bases. If you are looking at a eukaryotic genome, especially one with large introns like human genome, you might want to set max indel a lot higher. For bacteria, this is way more than enough. The next thing it tells you is the minimum score ratio that it uses, and this is based on the min ID that you gave BBMap. And then we have 200,000 pairs of reads. In mapping mode normal, that means that you are mapping Illumina reads and that you're not asking BBMap to align faster at the expense of quality. If you want to go a little more into that, the BBMap tutorial will show you what other kinds of settings you can use for mapping mode. Okay, so now it tells you a little bit of statistics about the mapping itself. I mainly look at this top part. So it tells you for the paired reads, right? What % of your read pairs actually map to the genome? And that's about 90% of them. That's really good, actually. I'm used to working with environmental metagenomics, and if I get 20% here, I'm so happy. It tells you the number of pairs of reads that mapped. And then if you multiply that by about more or less the length of a read, then you get how many bases actually mapped to your genome. And you have a very small amount of bad pairs or read that were thrown out for whatever parameter you gave BBMap to filter mapped reads by. Then for each read, you can see what percentage of the reads in this file were actually mapped to the genome, how many of them mapped only to one place in the reference genome, which is the ideal outcome, but there are always going to be reads that map to more than one place in a genome. For example, if you have some kind of a duplication, like more than 116S copy. This is how many reads were ambiguous. Ambiguous means they map to more than one place. And we'll talk a little bit more about that later. And then we had some low quality reads that were discarded. As you can see, this is a really good quality run, so very few reads were thrown out. Okay, now we talked about mapping more than one set of reads to the same reference. So for that, there is a really nice bash script called BBWrap. So once again, you give it the reference. And then where you would, if using BBMap, this is where you would tell it the name of the forward read. Now we're gonna give it the first forward read, followed by a comma, and then the second forward read, okay? And then for the reverse read, once again, you're gonna give it both of these reads, but this time you're giving both reverse reads. So make sure that you keep your reads in the same order. Otherwise, it's not gonna be able to find pairs of reads, right? If I reversed the order of these two files, then BBMap would fail. Okay, so once again, we can set the minimum ID. Actually, let's set it differently this time to see what that does. Another thing that we need to set is the type of mapper that we're using. So BBMap has multiple types of mappers. The default is going to be BBMap. But if you have PacBio reads, for example, then there are other mappers that are designated for that, because these reads are longer and a little different to map. And then finally, we're going to give BBMap two output file names. So we're going to have this one for the first set of reads. And then for the second set of reads, I'm just going to change the sample number. And now we can run this. And once again, it's going to take a couple minutes. So in the meantime, so one more thing that you can see at this point. When you use any sort of mapper, the first thing that it's going to do is it's going to index the genome. BBMap knows how to look for indexes that were already created. So when we started running BBMap earlier, here, the first thing that it did was it loaded the reference. So it created an index. It says no index available, generating from reference genome. Now, however, we already have the reference available because we did it last time. So now BBMap is going to use the same reference. And it says, well, it says that somewhere. Right, just loading reference and then loading index. It's not creating another index at this point. And this time, the minimum alignment score ratio is set to 0.816. Before it was about 0.9, right? So this time we set our minimum ID to 90% instead of 95, just to see if we get more reads mapping. I don't know. We should. So this is our first set that was already mapped earlier. And this time we have 94% of the reads that were mapped. Last time it was 92 or 90, no, sorry, 90%. We had like 3.5% more reads that actually mapped to our genome. Okay. So that was BBWrap. This is if you have one reference and multiple read files. Now the next thing we're going to talk about is what if you have multiple references. So for example, you assembled mags from a metagenome, and then you have the reads that you assembled from, and now you want to map them to your mags, to your collection of mags, to get an idea of their coverage, their relative abundance. So for this, there is a script called BBsplit. So the thing that BBsplit does differently from BBMap is this part right here. If you have a collection of genomes, you're definitely going to have reads that will map to more than one genome in your reference set. And the reason is that there are just conserved areas in genomes, right? And there are genes that are conserved. So what BBsplit will do is it lets you decide what you want to do with these reads that map to more than one genome. And you can use several parameters for that. So you can either pick the best hit and that's it. So a read is only going to map to one genome. You can choose to just toss all the reads that map to more than one place, and that depends on what you're looking for. So for example, if you're only interested in genes that are very variable between genomes, then maybe, I'm sorry, then maybe you don't care about the conserved regions and you don't care about reads mapping to them. Another option is you can save all of the places that a read can hit. So this is sort of a scenario in which a lot of your genomes are probably very similar, and you don't know what is the true place where this read is supposed to map. So one option that you have is just save all of them and assume that it just mapped everywhere. And the last option is sort of a hybrid, which is to write reads that map to more than one place into a separate file. And then you can choose whether or not to use them. Okay, so in this case, I don't really need to use BBsplit. So I'm going to skip it for now. But this is actually pretty useful. Personally, I really like this option. So I would, for my work, probably use unambiguous to best. So only pick the best hit. But once again, it's really up to your downstream analysis. Okay, one of the really nice things about BBMap is that other than giving you a BAM file, it gives you all sorts of textual outputs that are really, really useful to have. So let's look at a few of them. I'm going to run the first command again. So what I'm doing now is I'm telling BBMap that except other than the out_m output, which is the BAM file, I also want to look at these types of outputs. So CovStats gives you a summary of coverage statistics to your reference. And in this case, because it's just one single genome, it's not going to be super informative. But for example, if you're mapping to a set of genes, then you're going to have the coverage and all the statistics per gene. And the same would go for contigs. CovHist gives you data that you can use to build a histogram of what is the most common coverage for your genome. And we'll show that in a second. There is another type of output called base_cov, which gives you the coverage per base in the genome. Of course, that's going to turn out to be quite a large file, so I'm not going to do it here. But let's look at our stats. So here's what you would get. You would get the ID. So in this case, it's just one chromosome. But if you have multiple genes, then you're going to have multiple rows. And for each one of them, it's going to tell you the ID of the gene, however you provided it. So probably the header of the FASA. Then it tells you what is the average fold coverage. So in this case, it's about 7.8x. What is the length? So in this case, it's the length of the entire genome, which is about 3.5 megabases. But again, if you have a set of genes, then it's going to look at the length of whichever gene. The next thing it tells you is the GC content. Then what % is covered? So this would be like the breadth. How much of the genome in this case is actually covered by reads? And normally you don't want to look at things that have a low breadth, and that's going to be probably covered in the metagenomic lessons in the future. Okay, and then it gives you statistics like median fold and the standard deviation. And this is all really useful, especially if you take information about per gene coverage into R to play with it with data frames. So basically you already have a file that's ready to go to play with it. Now for the histogram information, let's see what that looks like. So here's what it does. It gives you a 2-column file. The left column is for coverage, or X, right? And then on the right column, you have how many bases are covered to that level. So 17,000 bases have 0 coverage. If we think about the fact that this genome is about 3.5 million bases long, this is actually a really, really small number. And then it looks like, once again, most of our coverage is here between 6 and 9x. And if you remember, the average fold coverage was about 8, so that makes a lot of sense. So if you wanted to create a histogram of coverage in this genome, you'd have the data already kind of ready. Okay. So the last thing that I'm going to go over, which is a little more complicated, is we talked about using BBWrap, right? I'm going to copy it here again so you can look at it easily. But we were using BBWrap like so, when we had multiple input files or multiple read files. But this gets really, really complicated once you have a lot of read files, which is usually the case. So if that's the case, then you could write a loop in Bash that will go over your read files and feed them into BBMap. So you don't have to sit there and write, I don't know, like the last project that I was working on had 48 paired read files. So I'm not going to sit down and write 48 comma separated file names. Could, but I'm not going to do it. So this is how I chose to do it. What I did was I defined a variable and I called it current directory or curdir. It's going to be set to data. So data is where all of our data files are, right? Okay. So now I can refer to this variable and it's going to go into this directory. And then I'm going to go through a loop. So the way you write a loop in Bash is very similar actually to how you would write it in R. So for every F, so file in this case, I like to call them F, in the current directory. And then out of that, I only want the files that end with 1.fastq.zip. So these would be the forward read files. Okay. Now you have to tell it what to do. So you write do, and you don't have to keep creating new lines. I just think it's easier to see what I'm doing this way. So the first thing that I'm going to do is define the reverse read. And the way I do it is I define F2 or the reverse read. And you can call it whatever you want. I like to call it F2 because I think it's short and easy to understand. The way you manage a variable within a loop in Bash is you write this $sign and then everything in a parentheses. And now you can write commands as if you were writing in a Bash terminal. But if you don't do this, then Bash is not going to know what you want it to do. So what I'm doing is echo F, so basically print the current file that we're looking at. And then what I want to do is replace the suffix of the file name, which is going to be 1.fastq.zip, into 2.fastq.zip, because this is how my forward and reverse files are represented. So forward reads end with 1.fastq.zip and reverse reads end with 2.fastq.zip. So this is all I did, is I defined the reverse read file name. Okay. The next thing is I'm going to define a base for my output file names. Now, if you think about how I defined the base before, I was using the sample name to do that. You can define this however you want. I am defining a new variable called fbase. Once again, $sign parentheses, echo F. And then I am using the command cut with the delimiter slash. And I'm getting what's after the slash. So F is going to also include the directory name. So really, F is data slash srr whatever fastq.zip. I don't want to use data slash in my base name for all of my output files. I just want to use a sample name. So I'm cutting out that first part, the data slash, and then I'm throwing out everything after the underscore. So if you look at the file names, we looked at them before, they look, like this, right? So we have the sample underscore 1 for forward, and then this. All I want is this first part. So I'm throwing out everything from the underscore and on. This is all I'm doing here. Okay, so now I have a forward rename, a reverse rename, file name, and the base name for my output files. And what I'm going to do is I'm going to run BBMap. As a reference, I'm going to use this reference file that's within the current directory. Now, you don't have to, if you're always referring to the same directory, you can just write the directory name. So in this case, it would be data. But if you want this to be useful for future projects, then all you have to do is change the current directory name. Instead of going through the entire loop and changing it in every single place, I think this is useful. It's really up to you. So I'm giving it the reference. The forward read is going to be, again, $, and to tell it to actually read F as a variable and not just write F as the name of the input file. You have to give it the $, and then these curly brackets. Reverse read is going to be $, curly brackets, F2. And now you're going to give this loop a bunch of output file names. So I'm using for all of them this structure, $, curly brackets, current directory, $, curly brackets, F base. And then this is going to be the covstats, right? So I'm going to finish this with underscore covstats.txt. So really it's going to give me a file that's going to go in the directory data, and it's going to be called srr whatever this underscore covstats. And the same for covhist. And just this time I'm going to go also and generate per base coverage, just so that you can see what it looks like. And then finally, the mapped BAM file outputs are going to be in the current directory. They're going to be called the base name underscore map.bam. So now all I have to do is write done and done. And now BBMap is going to start looping over, in this case, just 2 files that end with fastq.zip. And it's going to map both of them to the reference and create all of the output files that I requested. So it's going to take a couple minutes again, because it's mapping to a couple of genomes, but at least doesn't have to generate the index, right? Once again, we have the index already done. And then this time I set minimum ID to 95% again. So our minimum alignment core ratio is once again up to 0.9. So BBMap is a really convenient tool, or it's a set of tools along with BBSplit and BBWrap. And really my favorite thing about it is the fact that, for me at least, it's a lot easier to understand a %ID match of a read to a place in the genome than some, I don't know, some amorphous calculated ratio or map Q like in Bowtie that I don't fully understand how it was generated. And I can't fully understand it because it depends on so many different things, like the read quality and the scoring matrix. And I'm not supposed to understand how it works exactly, because it depends on so many different things. It's an algorithm. It's not a simple addition. So that's fine. It's just, for me, easier to understand. And I think it's also easier for readers of your papers to understand what minimum ID is. So this is going to be done really soon. And then we can go ahead and look at our output files. And once again, this is a binder, so it goes a little slower because I'm only using one mapping thread. But if you're using your own computer, you probably have at least 16 threads that you can use. It's going to go a lot faster. But even so, it's a pretty fast aligner. Okay, that's it. It's done. So let's look at our output files. So we now have 2 bound files, right? Oh, sorry. This one and this one for each sample that were just created. And then for each one of them, we also have the per base coverage, coverage statistics, and coverage histogram. So just before we finish, let's really quickly look at per base coverage and what it looks like. So it just goes position by position in your genome and tell you what is the coverage there. So if instead of the head, I'm going to use the command less, so I can just scroll through the genome. You see the coverage is starting to increase here, right? So it's really the end that isn't well covered. It's not the middle. I'm not going to go through 3.5 million bases now to show this to you, but again, if you're using R or Python and you want to plot your coverage per base of the genome, which is a really common thing to do, then this file lets you do it really easily. Okay. So I hope you enjoyed this tutorial as much as bioinformatics can be enjoyed when you're just starting up. And I hope it's helpful. And I'll see you soon in later tutorials. Bye.